nitrogenous or nitrogen bases of each nucleotide. And here's the thing, and this is, otherwise this wouldn't work. Guanine base pairs with cytosine and adenine with thymine. Apples grow on trees, cars go in garages, and that's just the way it is. Okay? Unless some mistake occurs and we get a mutation. But otherwise, this is the way it is. Okay? So, sides of the ladder, one strand, another strand, and then the steps connecting them, making it a double helix. Okay, there's a lot of information about this double helical structure, about in more detail about DNA, but we are not going to go into it. I want the simplified version because there's just too much. Again, enough for a whole year, whole life's worth of study. So we're going to simplify it. In fact, when I draw something on the board, you're going to see just how simplified it can be. In order for two new cells or offspring, if that's the case, and we'll talk about mitosis and meiosis later, a couple of weeks from now, DNA has to be replicated. One double helix results in two double helices, and they are identical. If we go back here, whoops, too far. I know you, well, actually, let's go this far back. So this is one of those items in what is showing as a chromosome for a karyotype. And this, the top, is one sister chromatid, and the bottom is another sister chromatid. If you couldn't see that, let me do it with this pen. This across here, across the top, is one sister chromatid, and this across the bottom is another. They are replicated double helices. They are identical to each other, and we're going to talk about that on the board. So let me get us back here. So this DNA has to be replicated, okay? It's the duplication of the DNA prior to cell division, okay? And our original strands, remember it's two strands. So this one and this one, this includes the bases, serve as templates to make the two new strands, making the two new double helices. Again, I want to put that on the board for you so I can explain it better. There are enzymes involved, like DNA polymerase. Someone asked about polymerase. Polymerase, poly, is just adding new nucleotides to each of the strands. But we're not going to worry about learning the enzymes. You can do that if you take a cell biology that's for majors. Before we get to replication on the board, I want to go on and talk about mutations. So mutations are errors or mistakes that are made with the DNA, and they could be simply a substitution. So that instead of having C here, see how these are in threes? These are actually whoa. These are the amino acids that are coded for in the recipe of the DNA, okay? And you can see that we have, you don't have to know this. This is proline right here, and CCT is saying, hey, that's the ingredient that goes right here. That CCT, which is cytosine, thymine, along, adjacent to each other. But what has happened with the mutation is that the C has been replaced, the cytosine, with a guanine, a substitution. And look, now we have a different amino acid. And that can be enough to make a completely different protein. In fact, hemoglobin A is normal hemoglobin. It's what most of us have. Someone with hemoglobin S, either some of their hemoglobin is hemoglobin S, or they've got sickle cell anemia or sickle cell disease, and they have only hemoglobin S. It's because of one amino acid that is different from when you compare hemoglobin A to hemoglobin S. That's how big the consequence can be, okay? From not having sickle cell anemia to having sickle cell anemia. Then there can be deletions, there can be additions, there can be what are called translocations. There are all kinds of mutations, multiple mutations, more than we need to discuss. How do mutations occur? Some of them are spontaneous. It's just an accident during replication. A lot of those get fixed. There's a lot of DNA repair. The younger and healthier you are and less exposed to certain mutagens, the more likely you are to be able to repair, your body repairs the DNA so that the mutation doesn't persist. So those are spontaneous, they just occur. Sometimes they don't get fixed. We'll talk about beneficial and deleterious mutations here in a moment. Sometimes there's something that causes the mutation. It's an induced mutation. And that is when mutagens are involved. And mutagens can be chemicals, like some of the chemicals we put out in the environment that then end up causing cancer clusters in different areas. Some of them are physical phenomena like UV light, which causes skin cancer because of DNA mutations. That's where the thymine dimers come in. Two thymines that are adjacent, like there'd be two T's together, instead of forming a bond with the other side of the chain, or the double helix, they form a bond between each other, and that's caused by UV light, and then that messes up the DNA. If it doesn't get fixed, then it can lead to, in our cases, skin cancer. So, really an issue there, mutations. But mutations can be beneficial, mutations can be deleterious, or they can be neutral or silent. So beneficial mutations are those that occur, whether they're spontaneous or induced, but they occur and the protein is better. It provides an advantage to the individual. And by doing so, with evolution, which we'll talk a little bit about later, it may become really common in the population, that particular mutation. These have happened throughout. This is one of the ways that this is a source of the changes that can lead to evolution. So this has happened all along. A beneficial mutation, you might not think of hemoglobin S as a benefit, but for people who live where malaria is a problem, those that they got the sickle cell hemoglobin from one parent but normal hemoglobin from the other are more likely to survive malaria. And before we started aggressively preventing and treating malaria, and even when we were doing what we thought was aggressive, 1 million people a year around the world, and they were mostly children under 5, were dying from malaria. Malaria was a huge killer. The numbers have come down to under 500,000, and that is amazing. But it took a lot of effort to get to that. But there are people who have a natural, not really immunity, but an ability to survive malaria compared to others. There are people who have some kind of genetic mutation that are more likely to survive AIDS. So, and it's not just in humans. These beneficial mutations occur in all kinds of organisms. And sadly, antibiotic resistance is a beneficial mutation for the bacterium. Not good for us, but good for them. Deleterious, or bad. These are mutations that cause problems, like the skin cancer, like lung cancer, like cystic fibrosis. So they cause a change in the protein, and sometimes it's so overwhelming that the organism doesn't even develop. Sometimes the organism is born but dies. Sometimes the organism lives a while but is more susceptible to different issues that occur, daily issues. So, and I'm saying organism because it's not just humans. We'll talk more about some of these deleterious mutations when we talk about genetics. And then some are neutral or silent. So basically the DNA changes, but the amino acid stays the same. When we talk about transcription and translation, I will explain how that can happen. So hopefully I have answered most of the questions except those about, hey, replication and how do we know it's semi-conservative? Well, that is what I'm going to show you next, but I need my the whiteboard, it's not my whiteboard, the whiteboard to do that.